Good evening, everyone. I am Willie E. May, Vice President for Research and Economic Development here at Morgan State University. We will explore the processes used to develop vaccines and the design and conduct of the clinical trials that are used to determine their efficacy and safety for the large populations that the vaccines will be deployed to protect. During the seminar and the focus group discussions that follow, we hope to address concerns being raised by the general population, but especially by the Black community who's been hit the hardest by this pandemic about the efficacy of the COVID-19 vaccines that have been developed at an unprecedented warp speed. Dr. Willie Vann will give the first lecture that will be focused on how vaccines are made. Dr. Vann is chief of the Laboratory of Bacterial Polysaccharides within the Office of Vaccine Research and Review, Center of Biologics Evaluation and Research at the Food and Drug Administration. He did his undergraduate work at Morehouse College, where, by the way, he was Phi Beta Kappa, and received his PhD from Purdue University. He did a postdoctoral, he conducted postdoctoral research both at the National Institutes of Health from 1973 to 1975 and at the Max Planck Institute for Immunology in Freiburg, Germany from 1977 to 1979. With respect to vaccines and their development, Dr. Vann is well published and has given lectures and chaired conferences and sessions within conferences with around the world. Dr. Margaret Bash will give the second lecture that will focus on the clinical evaluation of vaccines. That is, how clinical trials are designed and conducted. She is a medical officer with the Food and Drug Administration, where she conducts regulatory review of the design of clinical trials, looks at safety and effectiveness data, and the chemistry, manufacturing, and control information for investigational as well as licensed preventative diseases, at least vaccines, my bad. She is board certified in both pediatric infectious diseases and an affiliate associate professor in the Department of Pediatrics at the Uniformed Services University of the Health Sciences. Dr. Bash leads a research program focused on the antigenic diversity and functional immunoassays of pathogenic Neisseria, and Dr. Bash, you will have to correct my pronunciation when you speak, to facilitate the development of vaccines for these bacterial pathogens. She received her undergraduate education at Colby College in Maine and received her medical degree from the Uniformed Services University of the Health Sciences while on active duty in the U.S. Army. Immediately following the two lectures, Dr. Kim Sitnor, Dean of the Morgan School of Community Health and Policy, and Dr. Fareen Kamingar, PhD, MD, who works as the Morgan Assistant Vice President for Research Administration. The two of them will facilitate a focus group discussion regarding many of the concerns that people of color are expressing about taking these vaccines that are being developed and deployed at unprecedented speed. So our first speaker will be Dr. Willie Vann. Dr. Vann.
All right, I've unmuted myself. I should go with share screen or what? Okay, share sound, share. Let's see, minimize this. Here we go. So good evening. I wish to thank Morgan, especially Dr. May and the Dean, for inviting us to give this talk on vaccines. Margaret and I work at the Laboratory of Bacterial Polysaccharides, where we regulate and do research on vaccines that are bacteria that cause disease and that are covered with polysaccharides. So we are primarily interested in bacterial vaccines and polysaccharide-based bacterial vaccines. As Dr. May mentioned, there are two lectures. The first one will be on vaccine manufacturing, and the second one will be on clinical trials. I will talk about how bacterial vaccines are made, specifically polysaccharide vaccines are made, and how they are generally developed. Some of the principles of making polysaccharide vaccines are generally applicable, and some of the approach that is taken to monitoring and ensuring consistency and potency of these vaccines are generally applicable. But to give you an example of how things are done, and based on what we know most about, I'm going to talk about polysaccharide vaccines. So I must start with this disclaimer, and that my comments are an informal communication and represent my own best judgment. The information presented here does not bind or obligate the FDA. We are not speaking for the FDA. We're giving you our opinion and how we judge things work. So where do vaccines come from? So every vaccine that is licensed or that is being considered for licensure originates as an idea in a laboratory. They're science-based. They originate as ideas developed in a laboratory and in a library. The ideas are based on years and years of literature and science that has been developed that gets applied to the development of a vaccine. To illustrate this, I'm going to play you a short video of a pioneer in vaccine development who describes very quickly how he developed a conjugate vaccine for Haemophilus disease. Haemophilus was a disease that was very prevalent, but the development of this conjugate vaccine actually drastically reduced the disease. Pay careful attention to what he says. What will happen later in the talk, I will refer back to, but pay careful attention to what he says. He illustrates how vaccines come from the laboratory, the library, and etc.
About that time, the pneumococcal polysaccharide vaccine was developed, and because I came back to my university thinking about polymers, I decided I'd work on Haemophilus, and I was soon joined by people at Harvard University, David Smith and Porter Anderson. We first purified the polysaccharide and then did the usual tests to show that induced antibody had biologic activity. We studied it at Mecklenburg County, North Carolina, and we soon found out that it didn't immunize infants and children, that age group with the highest attack rate of disease. It did induce some antibodies about the age of 2, and it was used as a vaccine for a while, but there were too few cases of it, and it wasn't 100% effective or even close to it. We then read articles by the very famous chemist Avery. Avery was the first man of Avery, McLeod, McCarty, who discovered DNA. He was one of America's great scientists. And we made vaccines by covalently binding the polysaccharide to a protein. And when that happened, it conveyed some of the immunologic properties of the protein to the polysaccharide. And now this so-called conjugated polysaccharide or conjugate induced antibodies in infants and children. And very quickly thereafter, 3 or 4 major manufacturers made conjugates and quickly showed that it was almost 100% effective. About that.
So this is how you get the ideas and how they actually are converted into a vaccine that actually goes into people. So here on this slide, I've briefly outlined the process, how they get manufactured. You come up with an idea and a concept in research in the laboratory. And then once you realize that that's going to work in animals, it's time to put it into people. So it goes into development where you have a pilot production. You scale up from small laboratory production to pilot production under conditions that can be used in a clinical trial. And this is the stage at which clinical trials begin, which Dr. Bash will talk in detail about. Clinical trials are conducted based on materials that are produced in the pilot plant and also material that is actually produced at commercial scale. The results of the clinical trial are submitted to the regulatory agency. If everything looks well, the vaccine gets licensed. And then it goes into commercial scale production. And that commercial scale production can last for many, many years. So Dr. Robbins talked about polysaccharide conjugate vaccines, and this is what we work on. So let me start by introducing you to what these vaccines really are, so you know what we're talking about here. So many pathogenic bacteria that cause diseases such as bacterial pneumonia, meningitis, otitis media, urinary tract infections, typhoid fever, etc., have surfaces that are coated with specific polysaccharides. These polysaccharides are really just chains of sugars. And to the bacteria, they act like a moat or the walls of a medieval castle, that they protect the bacteria and ward off the attack of the host's immune system. These polysaccharides are very specific chemical structures. So you can raise antibodies to them, and you can use these antibodies to identify the bacteria that are causing disease, the specific strains that are causing disease. And what people have learned over the years is that you can use these polysaccharides without having the whole bacteria, just isolate the polysaccharide, and you can develop a vaccine. And that happens because if you raise antibodies against those polysaccharides, the antibodies will protect against the particular bacteria that is coated with that particular polysaccharide. So these polysaccharides have been vaccine targets for prevention of disease for years. These surface polysaccharides are used as vaccines in either of 2 forms. You isolate the polysaccharide itself, you purify that, and that becomes your vaccine. You develop that and produce that, and that becomes your vaccine. Another way to do it is actually to isolate the polysaccharide and you couple it to a protein. And as you heard Dr. Robbins talk about, when you couple it to a protein, you actually change the way its immunity and its coverage. And that's a glycoconjugate vaccine. Most of the vaccines that are polysaccharide based now are glycoconjugate vaccines. And there are lots of them. And this table is a list of the vaccines that have been licensed in the United States and Canada that are based on polysaccharides and against disease. Many of these vaccines you might recognize or have seen in a commercial. These vaccines are very effective. And to demonstrate that, there's a particular slide that I like to talk about, and that's about the meninge A. So up until 2010 or 2011, sub-Saharan Africa was actually plagued by epidemics of meningitis against group A meningitis. And an organization called PATH that is funded by the Gates organization used technology that was developed by scientists in the laboratory of bacterial polysaccharides to couple, to conjugate the MNJ polysaccharide to a protein. And that vaccine actually was introduced into Mali, Niger, and Burkina Faso in 2010. In 5 years, so in 2015, after immunizing over 250 million vaccinees, not a single case of group A meningitis was detected. As this slide shows you, that back in 1996, 1997, the epidemic had a result in 25,000 deaths. So these vaccines can be extremely effective. So why conjugate vaccines? So while polysaccharide vaccines are immunogenic and they protect adults, they don't do a very good job of protecting infants, and in adults, the immunity wanes. So conjugates actually work much better and they dramatically improve immunogenicity, so you can cover all age groups. So what do you do when you make a conjugate? You isolate the polysaccharide from the surface of the bacteria, couple it to a protein, and then you chemically couple these two, and what you end up with is a complex mixture. This complex mixture, as I said, works very well, but it's very complex. And what happens is this complexity introduces manufacturing and regulatory challenges. Some of the uncertainties that it introduces is you get all of these molecules that you've made by coupling, and you don't know exactly how many are in there and the structure of each one that's in there. The other thing is since there's so many of them, you don't know which ones are immunologically important. Some of them actually may just be spectators and not doing anything, and some of them may be players, and you don't know whether the players are all in the audience or not. And the other thing you don't know is how do you control the synthesis of these structures? So there's one thing that actually is very important about making a vaccine, about making all vaccines, and that is they have to be made consistently. Since you only do a clinical trial for a particular vaccine once, the vaccine batches that are manufactured years after licensure are expected to have the same effectiveness and safety profile as the vaccine batches that were tested in the clinical trial for licensure. So the other thing is the life of a vaccine, regulatory life of a vaccine is very long. We're talking here about development, which is actually the portion where you come up with the idea and you put in clinical trial and you do a clinical trial, then you license it. Then after that, even up to 20, 25 years later, you still have this vaccine and it actually has to be regulated and you have to control it. And the way it's controlled is by characterization of its manufacturing process and characterizing the vaccine itself. The goals of the characterization are to ensure the consistency of the product potency and the purity, to ensure that it's safe, so that it has the same safety profile, and you use the characterization actually as a tool to assess stability of the vaccine, because it has to be stable as long as it's on the shelf. So what happens is critical parameters for manufacturing process and the vaccine components are established based on what the current understanding is of the effectiveness and safety. And these properties are used by the manufacturer and the regulatory agency to monitor the process and the vaccines throughout the manufacturing life and throughout the storage of that particular vaccine. So what are some of these things? So this is an example of, I don't expect you to read all of the slide, but this illustrates you the kind of detail that is looked at for a conjugate vaccine. And this is a cartoon of a conjugate. And you can see there's all kinds of things that actually have to be looked at and monitored to make sure that the vaccine is actually performing correctly. Next slide. Okay. Not only do you look at the active ingredient, you look at all the components that go in there. You monitor the process. You look at the starting materials. You look at how the intermediates are activated. You look at proof that actually you got a conjugate. You measure the conjugate and you measure how much did not get conjugated. There are reaction byproducts in the chemistry. So you look at potential reaction products. And then you know that certain properties have to make it work when it's in humans, and you look for those properties. So once you've got the final product, this is what and it's going into the vial. You also want to characterize that final product. You want to make sure that what you think is in the vial is actually in the vial. You look for have an identity of the components. You look for the important epitopes that are supposed to be there, that are supposed to introduce immunity. Look for the amount of that conjugate, which is the active ingredient. The amount of free saccharide in there, which is actually giving you an indication of stability. And also you have other measures of stability that you look at the final product. That's before it actually gets injected into people. The other thing that's important is actually setting specifications for not only the product, but for the intermediates, for the drug substance, which is the active ingredient, and for the drug product. The goal is to ensure that the consistency of manufacturing, the product potency, and safety are always there. These specifications describe the acceptable limits of results from critical assays used in the characterization of the manufacturer and the stability assessment. These specifications are derived from the manufacturing experience during development and clinical trials and the experience at licensure. And that's actually, it's important that actually all of these experiences are used, and especially since things are manufactured on a different scale, on various scales, by the time you get to commercial production. So one of the things that actually is actually something that actually has to be dealt with is setting specifications based on product capability. Most of the time, when you do a clinical trial, you're using only a few lots. You might manufacture 3 to 5 lots of vaccine, and they may be manufactured in a different way. And the ones that actually you're going to use for the final clinical trial, you maybe only have about 3 lots. But 3 lots is not enough to actually give you good statistical data for specifications. So there's a way you actually have to approach that. And that the thing is, the question you have to ask is, how closely can you manufacture this to the same lots that actually you used in the clinical trial? Because you want what you make later in life to mimic what's in the clinical trial. So what you can also rely on rare occasions, and because most of the time this doesn't happen, but sometimes there are vaccines that don't perform as expected. So you can look at those lots and say, okay, this is what we don't want it to look like. And you can actually glean some information from the properties of those types of vaccines. And you can look at the average of what you've been able to make and what you are able to make later. So the other thing is, you always want to strive to have sufficient manufacturing experience to set specifications. So you may have interim specifications, but later on, after you've made a number of lots and it's been licensed, you can tighten those specifications up. This is a very important part of regulating a vaccine, which is actually something that's struggled with by the manufacturer and by the regulatory agency. The other issues that you have to deal with once you've actually licensed the vaccine that's out there is process changes. The product that is licensed is a process. It's not a specific something in a vial. It's the process of getting that into the vial. And once you change that process significantly, you've changed the vaccine. You can change it significantly, so you actually have a new vaccine, which actually means you got to do a new clinical trial. So this is something that actually happens throughout the life of the vaccine, and that is to figure out, how do you deal with process changes? Say, for example, company decides that they're selling a lot of it, they would need a new factory, so they build another factory in Colorado, where they actually used to be making it in Baltimore. That's a significant process change. We want to know is, does that change affect immunogenicity? Does it affect safety? There are other changes in the process that actually happen. We have to go back and actually evaluate what's happening in the vaccine. We have to go back to the library again, look at the scientific literature to see what kinds of things that actually could affect the vaccine, and do these changes, science say that these changes will affect the product. One thing that actually helps a lot in evaluating changes and even in setting classification is in product development. Excuse me. This is really the experimental part, the industrial experimental part of developing a vaccine, where lots are made under various conditions, and you end up with a vaccine, with something that actually you eventually put into the clinic.
10 minutes.
Okay. The experiences that you actually learn there actually inform you on what kind of changes you can make and what affects these parameters. Important to remember that for biologics, for all biologics, the process is the product. And when I talk about changes, there are many unexpected things that actually happen with things that are actually seem to be trivial. And I've given a few examples here that were taken from things that we actually saw. So, for example, a company changed the way they purify the polysaccharide to increase purity. This had dramatic effects on the analytics. It didn't pass specifications, or the specifications started trending differently. Or they changed the process by decreasing the salt concentration when they were purifying the carrier protein, or actually coupling the carrier protein. That really affected the chemistry and affected the specifications. The raw materials, actually, or other things, the containers that these things are stored in, even the vials that actually, the glass vials that the vaccine is in, can actually dramatically affect stability. So if you change from a vial that's one type of glass to another type of glass, you actually have to look to make sure that you haven't altered the product, and you haven't changed its stability, and you haven't changed anything that affects its potency. Change in the scale of conjugation or any scale of manufacturing can actually have dramatic effects. You're changing people, you're changing places, you're changing equipment. Another thing that seemed rather trivial was when the manufacturing process was changed, it led to a slight increase in residual metal in another type of vaccine, actually resulted in some drastic changes, because these metals, trace metals, actually turned out to be catalytic. So the regulatory agencies and the manufacturers pay very careful attention throughout the life of the vaccine to all kinds of changes, and any kind of change that is made to a vaccine process actually has to be submitted to the regulatory agency for approval. It gets evaluated as to whether it affects the potency or the safety or the purity of the product. So I want to thank you for your attention. To a shout out to my mentors, and to thank the management at CBER, the review teams, and my research staff. And that's it. I think I'm ahead of time.
Okay, we will withhold questions until after our second lecture by Dr. Margaret Bash, who will describe how the clinical trials that determine the efficacy and the safety of these vaccines are carried out. And she will comment on the safeguards that are associated with that process to make sure that these vaccines are efficacious for a diverse populations. Dr. Bash.
Thank you. Thank you, Dr. May. Can you see, is my slide? Oops, no, share. Well, thank you very much. I'm really appreciate the opportunity to be able to share with you some of the information behind what goes into evaluating vaccines in the lead up to licensure and to a certain degree following licensure regarding their safety and effectiveness. I have the same comment in terms of disclaimer. We're here to provide general information about the process, which I hope is very useful. When people ask me, I say vaccines are safe and they work. And I do truly believe that. I've spent most of my career in the field of vaccines. But I don't say that from the perspective of, I say that, so just trust me. I say that because I really feel it's been part of my work and the work of the people at FDA that I work with to be trustworthy in the way in which we assess these vaccines when the information comes to us. And I hope that through the process of this talk, you'll have a better understanding of what that process is, but also where you can go to find more information, where questions are, so that you can develop a sense of confidence or identify the questions that you have that you need answered in order to be confident about the vaccines that are available. So like most government agencies, we are driven by applicable law. And for us, that means the Public Health Service Act. And so by law, anything that is licensed to be used in diagnosis, cure, mitigation, treatment, or prevention of disease. In other words, if it has a label, it's not a vitamin. If it has a label that says this will do this for a certain problem, that means it's a drug or a biologic or a vaccine. And it has to actually be shown to be safe and pure and potent before it's licensed. I like to talk about this pure part, which is what Dr. Vann just described. And in my very simplistic way, I talk about this as grandma's apple pie. I'm sure everybody has an example of something that no one else in the family can make except one person. And if it's made by somebody else, or if it's made in a different place or at a different time of the year, you don't get the apples from the same orchard, it's not going to taste the same. And to a great degree, biologics are the same because they're so complex, you can't just do a test at the end. And so those little changes are going to make them different. And as Dr. Vann pointed out, the main studies of safety and potency, which for vaccines, we're really talking about efficacy here, they're done during that last part of the development of the vaccine leading up to the licensure. And it's all those other tests that go on later that make sure that 20 years down the road, you still have the same vaccine that was shown to be safe and potent years before. So I hope to be able to step you through who is involved in vaccine development and licensure, what is the process that we use to license a vaccine, when do key events happen, where are vaccines studied, how do we know that they're safe and that they work, and why is this process important? So for the who, most of what I talk about will be the work that's done at the FDA, but also involved in the whole process of bringing vaccines to people. There are other players, and the CDC plays a big role in deciding the policy of how to use vaccines once they've been licensed. What's the most effective way to employ them within the country? Is it routinely recommended or just to particular populations. And both FDA and CDC have outside experts that serve on advisory committees to advise, to guide us. Certainly academic institutions, many of the initial ideas are developed in research at universities, and non-governmental organizations play a role. Industry is obviously a big player here. They are responsible for taking those good ideas that are looking promising and running through the development and bringing them to something that can actually be used in a community prescribed by your physicians. But also you and your communities are involved, both in the conduct of clinical trials, participation in clinical trials, concerns and questions and community input all play a role in bringing vaccines and to fruition. So what is done to study vaccines? Well, fundamentally, they're experiments. And we have experiments in animals and we have experiments in people. Those in people are called clinical trials. And there are various stages that we go through in clinical development. Preclinical is all animal work, but it lays the groundwork. It tells you whether you have a very promising candidate or not. In phase 1, the studies are very small and are very closely monitored and may be enrolled very slowly so that you see just a few people at a time, maybe at the lowest dose. It really, the way the phase 1 studies are done depends a little bit on how novel the new vaccine is and how much other experience we might have with similar kinds of vaccines. So these can go very slowly, they can be very small and very, very closely monitored. Based on that data, you can move into slightly larger studies of several hundred subjects and work out some additional details, really preparing for this phase 3 time frame, which is where you are going to really show that the vaccine works in the population that you intend to use it in. And that is generally done in thousands of individuals. Safety is monitored throughout all of these stages. Now, these all lead up to licensure, but you can't administer vaccines to people until they're licensed. So how do we conduct these studies before licensure? And that process is called an IND. That's an investigational new drug application. So anytime that a drug or vaccine is being administered in a clinical study before it's been licensed, it has to be done so under this kind of application that is submitted to the FDA. We review it and monitor the studies as they go on. We have regulations on this too, of course, and this is the 21 CFR Code of Federal Regulations. The basis, this should be a trivia fact for science people like us, but the regulation really seats in interstate commerce. And when an investigational drug is under an IND application, it allows this to be lawfully shipped across state lines for clinical trials. Well, what is our primary objective in reviewing INDs? First and foremost is to assure safety and rights of subjects. And we do that by reviewing the plans for human subjects protections through the institutional review boards or IRBs, as well as data monitoring committees. We review the consent process that's planned for that study, and we review the consent forms. A second big role is that we make sure that the study is adequately designed to permit evaluation of safety and effectiveness. In other words, depending on the stage, the study has to be able to accomplish its goals. Because if it really can't, if it's not designed well enough, or if it's not run well enough to generate data that's going to be useful, it's really not right for people to be exposed to an investigational vaccine. So it's our job really to make sure that the studies are designed so that in the end, they will provide the kind of information that we need to assess to decide on a license or not. We review these clinical trial protocols before the study is initiated so that issues can be dealt with before the study started. You really can't fix anything after they've already done the work. This includes trial design, methods, materials, the location, the statistical analysis that's planned. And we also look at the investigator's brochure. This is the body of information that is provided to the investigator that will be running the study at the local study site. And we want to make sure, does this investigator's brochure provide the investigator with everything that he or she needs to be able to answer people who are participating in the study? Will he be able to answer those questions and detect problems in the study if they start to arise? Since someone who volunteers for a study would be dealing with the local investigator, not somebody at the FDA, it's really important that these investigator brochures have the kind of information and detail that that's necessary. There's unique safety considerations in the context of preventive vaccines. So in the regulations, the definition of safety is specifically put in terms of in relation to the condition of the recipient. And we always keep in mind that for vaccines, the target population is often millions of healthy people, including young infants and children each year. And that sometimes state governments can mandate vaccines for children attending public schools or daycare centers. And that each individual's risk for some of these diseases might be quite low. The risk might be sufficient in the whole population that vaccination is important to continue, but each individual's risk is not very high. So for those reasons, the safety that's necessary for vaccines is really, really high. We have a very low tolerance for vaccine-associated risks. It's also important to know that when a vaccine is likely to be incorporated into a routine vaccine schedule, like the routine infant schedule, those phase 3 studies are done in the context of all the other vaccines that are routinely given. So the studies really do look at the use of that vaccine as it will be used after licensure. When do key events happen in vaccine development? Well, I've been talking mostly about this process that happens under the IND, and when all of this is completed and the data are pulled together, and assuming that they are favorable, they will be compiled into a biologics license application. But more often now we see that there are many times questions even before clinical trials start that might influence these clinical trials. And so we have a process for sponsors, manufacturers to ask us questions even before they've submitted an IND. And we also have an influence on things that happen post-marketing. And these are called phase 4 studies. Where is clinical data generated? Well, it may be in the United States or internationally. It can be in healthy people, populations at risk, populations with specific conditions. We do clinical studies, or people do centers, private doctors' offices, really all kinds of different places. And all of those factors might vary by the particular vaccine, the study, and the clinical developmental stage. But ultimately, by the time you get to the license application, the intent is to have data that will support the intended use. So in other words, we need the safety and the evidence that the vaccine works in the population that is likely to be using it after licensure. How do we know if a vaccine works? Well, we say the gold standard is a randomized controlled trial. And I've created a little cartoon here to kind of illustrate what that is. If you have a new vaccine to prevent a particular disease, it's given to some people, and then a placebo, maybe just saline, or maybe it's a control vaccine. It has nothing to do with the disease that this vaccine is going to prevent. You can do this either way, and that's given to some other people. But the people who receive one or the other are randomized, and in the best of circumstances, it's conducted in a double-blind way. And what that means is that neither the study participants nor the study personnel know whether a particular person got the vaccine or got the placebo or control. And that's important because then everybody is treated exactly the same as they go about their daily lives. And as they're being observed during the conduct of the study, the disease process that this vaccine is designed to prevent may happen, and some people may become sick. Well, those people receive appropriate medical care, but it's documented and tracked who has become ill with the disease. And at the end of the trial, those people who did become sick are separated into the group that had received the vaccine versus the group that received either placebo or control. And when people talk about vaccine efficacy, what they mean is what was the percent reduction? How much did you decrease how many times that disease occurred in a population of people by giving them the vaccine compared to how much the disease happened in the group that didn't get the vaccine? And that's how we measure efficacy. How do we know if a vaccine is safe? Well, safety data is collected at all stages throughout clinical development. And generally for a vaccine, there's a minimum of several thousands of study participants. We also get safety following licensure, either through post-licensure commitments that we agree on. These are called phase 4 studies. Sometimes there is registry data that's required of the sponsor. And also we have something called the Vaccine Adverse Event Reporting System.
10 minutes.
Theirs is a large data collect and analysis system that's co-managed by CDC and FDA, and it by providers, by hospitals, by manufacturers. It's very large. It's a passive system, but because of the expanse of the system and the ability to search for certain things, it provides a good way of exploring for unusual events and seeing whether there's any pattern of association with vaccination or not. The FDA recognizes and is working to improve and enhance diversity in clinical trials. It's important that clinical trials reflect the population that's most likely to use the drug or vaccine if it's approved. And recent guidance has provided some recommended approaches that could improve enrollment of underrepresented populations. The bottom line is that we want to study new products in the people who will be using them. And in order to do that, we need to enroll relevant populations with regards to age, sex, race, and ethnicity. This is mainly done because obviously participating in a clinical trial is voluntary and should be, and deciding to not participate in a trial is of course voluntary. So it's more a way of making sure that there aren't roadblocks to people participating, getting rid of unjustified exclusions, incorporating some things that may promote inclusion, and through that, hopefully providing some ability to get a broad spectrum of genetics, metabolism, diet, environmental exposure, many things that may affect how well and safe vaccines and products are. The new guidance really promotes including adequate numbers and, importantly, analyzing the data by race and ethnicity so that if there are population-specific differences in safety or effectiveness, those will be detected. It's now recommended that sponsors include a plan for inclusion of clinically relevant populations no later than the end of phase 2 so that that can be incorporated into those big pivotal phase 3 studies. So why is this process important? Well, vaccines save lives and they help people in communities stay healthy. Many vaccines have the greatest impact and vaccines are among the most cost-effective health interventions. By ensuring that vaccines are safe, pure, and potent, FDA serves public and advances public health. But none of that happens if they stay on the shelf. So I think that transparency and public trust are really critical to the success of vaccines in order for them to really achieve what they are capable of in terms of disease prevention. A couple of examples here. If we look back in last century when I was young, we had a lot of many of these diseases that essentially are non-existent now. The decreases that have been achieved through vaccinations are really remarkable. The vaccines for children run by the CDC projected that vaccination of children born between 1994 and 1918 prevent nearly 500 million illnesses, help to avoid nearly 1 million deaths, and saves nearly 2 trillion in costs. And this is also reflected worldwide. These numbers are yearly, every year worldwide. Millions of cases of measles, tetanus, and pertussis are prevented. So back to the vaccines are safe and they work. And that's what we mean when we find that the evidence is there for licensure. But what we really are looking at is how effective, how safe, and in whom. We characterize the local and systemic reactions and as well as adverse events, short and long term, often for up to 12 months in vaccine trials. And serious adverse events or unexpected adverse events are collected and evaluated for their relatedness to the vaccine. Effectiveness, we talked about the clinical endpoint efficacy type of study. Sometimes you can't do that. Either the disease isn't common enough, or there may already be a vaccine and now we have another vaccine, another manufacturer, and it's good to have that breadth of supply, but you can't have a randomized trial with some people not getting vaccinated. So in that case, we have to have a marker. It's usually a blood test, and we can use that as long as there's wide consensus and good supporting evidence that that marker does predict that the protection that the vaccine affords. It's important to keep in mind that the risk benefit, of course, is affected by how severe the disease that a vaccine's preventing and how common it is. So me, in my work, I'm a show me the data kind of person. And I think that we all should be that way. And one of the questions I think that's really important is how can you find out what data are available for a vaccine? When you get a vaccine, you're given something called a vaccine information statement. These are produced by the CDC, and they do explain the likely direct benefits and what you might experience from that vaccine, but they don't describe the background clinical trial data that these risks and benefits were established based upon. But that information is available, and both through the reviews that are done at FDA and through our advisory committee meetings. The Vaccines and Related Biological Products Advisory Committee meetings are public. There are video casts, there's transcripts available, and there's for And when licensing happens, the data that we've reviewed, our review of the analysis of those data, our interpretation of that, and our conclusions are all put into a memo. And those memos are also published online and available after licensure. Package insert. I mean, the little piece of paper that's in the box when you get your medicine, the type is so small you can't possibly read it. But they are also available online with where you can read them. And I just want to show the kind of detail that can be in there if people are interested. For one vaccine of a meningococcal vaccine, this one was studied in 3 different multi-center clinical trials, 9,000 infants. Here's the ethnic and racial diversity. Hippurix was studied in nearly 3,000 children in the US. Half were male, 61% were white, 8% Asian, 9% Black, etc. And the safety and effectiveness data are summarized in those tables. I wanted to end up on this just because I thought it was a good example. There was a study looking at awareness and trust of the FDA. This summarizes these for adolescents, teenagers, and most adults know the CDC and the FDA are there. A little bit smaller proportion trust them, but overall, we're trusted more than the general trust in the government as a whole. But I'd say this is barely a passing grade, and I'm kind of more of an A student type of person. So my goal really is for our information to be accessible enough and widely understood enough that there's good trust in our vaccines. I think it's really important. So I've put together some resources here. I think the FDA, CDC, clinical trials, all clinical. Importantly, coming up, there's been an ACIP meeting on the COVID-19 vaccine on the 1st of December. Thursday of this week, the VRBPAC committee meeting for the Pfizer vaccine will be taking place. And next week on Thursday, the Moderna vaccine will be discussed. And as I'd said before, these advisory committee meetings are public and broadcast, and they know there will be a lot of interest. So they've got YouTube and a lot of different mechanisms so that it won't overwhelm the system. And so I should move on. I'm over time, but I am happy to take any questions, as I'm sure Dr. Vann would be. Thank you.
Okay, thank you. As a point of order now, Dr. Kim Sitnor will moderate the question and answer session that pertains specifically to these 2 lectures. Immediately following that, she will be joined by Dr. Fareen Kamangar, who will facilitate a lively discussion, I hope, that will be focused on the COVID-19 vaccine that I'm sure all on the call are anxious to have their questions answered and to provide comments regarding. So Kim, take it away. I think there are 2 questions in the question box.
Yes, sir. And just again, thank you, Dr. Vann and Dr. Bash. I thought they were very informative presentations. I have questions. I'm going to act like an attendee at some point if I get a moment, but we do have 2 questions in the Q&A box. And the first one is addressed to Dr. Vann, and it is, are the Moderna, Pfizer, and AstraZeneca vaccines polysaccharide conjugate vaccines? It's a 2-part question. That's the first one.
The answer is no. Those 2 vaccines are based on messenger RNA, and they are, what happens is they get injected and your body actually uses the messenger RNA to make a protein which you immunize yourself with. So no, they're made They're different.
Okay. And the second part of the question was, is the process for constructing the vaccine, this is addressed to Dr. Vann, any different under the emergency use authorization process? So in terms of what you described, is it any different? And I think Dr. Bash may also want to answer that question.
No, no. The way a particular vaccine is actually made doesn't change when it's actually going from emergency use or license. In this case, they actually have to be made by the same process.
Okay. And then Dr. Bash, just because you described slightly different as emergency use authorization in terms of phase 2, phase 3 clinical, does emergency use authorization differ from your regular practice and processes? And if so, how are they different?
So the scrutiny of the clinical trials, not any different. So the process of looking at the preclinical data and moving into phase 1 and phase 2 and phase 3, each of those clinical trials was submitted and reviewed just as I described, quite, rapidly as could possibly be done, and ensure that the trial design was appropriate as I described, that it would answer the questions that needed to be answered about safety and effectiveness. Now, in this particular case, and I'm going to talk in general here because, as we mentioned, we're not going to be able to answer all the questions about the COVID-19 vaccines, but there will be a lot of discussion of this on Thursday. And there were discussions earlier, an earlier advisory committee meeting about emergency use authorization, because it was anticipated that if the vaccines really started to look like they were working, then you wouldn't want to wait too long. There's a balance here that, waiting too long, people are getting sick every day, waiting not long enough to gather enough information to be sure that you have found anything that might be a problem, or have a good idea that there's not a problem. So that balance was anticipated, and the trials will be continued as planned, but these trials were pre-specified with interim analyses that would allow a middle look. So like in that cartoon that I had, where you don't know who got the vaccine and who got the placebo, there was a separate committee that, after they had a certain number of cases, that separate committee could go in and pick that apart so that it would allow people to see if there was a good effect from the vaccine sooner than waiting until the very end of the trial. One of the things that was, I think, quite clearly expressed by the advisory committee is that it is still important to complete those trials. And I mean, the bad news about having so much disease circulating right now is, in a bizarre sort of way, means that these trials have been moving along very quickly because they're based on the accumulation of cases. And so that early look is what showed that there were a lot more cases in the individuals who had received the placebo than those that had received the vaccine. That's why it allowed them to bring that data in early. But the full process presumably will still go through that whole process. And safety is generally, emergency use authorization there's mechanisms to allow something that we think works, but you might not know exactly how well it works yet, because you might need more study data to know exactly how well it works. And there are mechanisms at FDA that not just emergency use, but other mechanisms too that allow vaccines to become available a little bit earlier, or other medicines to become earlier when there isn't anything else. So you've got nothing and you've got something that you know works, but you just don't know how well it works. That's the balance here, is to allow things that we know to work go forward. You can say, how well do they work? And all along, they really, the safety has to be there, or you kind of can't go forward.
Okay, thank you. So I think the general tone is that emergency use authorization does not undermine safety or any other processes. And it also was not created for COVID. But obviously this is something that is part of the general rule that can be applied. So I want to make sure that was clear. It was not created just for the circumstance. You already have these rules and authorizations in place. Okay, great. Next question from the chat is, can you help us understand about sample size, which is always a good question, I think, during phase 3 trials? Why 30,000? Is that a magic number in phase 3 trials in general? And I think this is addressed to Dr. Bash.
Okay, so this is a really good question, and it's not an easy question to answer, because when you design a study, so an important thing about a study is that you have to set up ahead of time what you're going to look at. You have to design the study ahead of time, because if you think about it, if you do a study and you haven't said what you're really going to look for at the end, you could go fishing around all kinds of endpoints and say, oh, look, I got something that looks good here. And that's not really fair. That might be what you do in the lab when you're first coming up with an idea, but it's not what you do in your pivotal clinical trials. But you haven't done the study yet. So you have to, based on that phase 2 data, you make a good guess. You make an estimate of how good do you think it's going to work? And how much would it need to work in order for it to be worth using? So, early on, I said we weren't going to talk about COVID, but I know that's in people's minds, so it's easy. There were reports that FDA said 50%. Well, that was more not saying that's what we thought the vaccine would be, but if you had a vaccine whose efficacy was even as low as 50%, it would still have a big impact across the whole population. So that's part of that planning. It's a very educated guessing. Like, based on your phase 2 data, you think, how many people do we need to have in order to be able to see this effect if it's there? Well, it turns out if it's an even better vaccine than you thought it was, you'll be able to see that probably with a smaller study size. So you may still want the 30,000 because that's the plan and those people were enrolled and we should follow them all for the complete safety data. But for a very highly vaccine, it's not unreasonable that you would be able to be certain it didn't just happen by chance alone, that it's this effective with a much smaller number.
Right, that was a great answer. I wish my students were on the line. I hope some of them are. So it's the real world reality of sample size and power, and why do you need these things, right? So that was a really
exactly.
Thank you. So we have another question. What are the ways that race, in particular, being African American, impact the manufacture? So I think this would be for Dr. Vann, the manufacture and the clinical trials for vaccines. So, we say we need to have diversity, and obviously we want enough African Americans, but I think the question is saying why? What impact does it have on the manufacturing process and the clinical trial itself?
On manufacturing, it doesn't particularly, except, for example, well, I can take the example of the vaccines in Africa, and there you actually have it's socioeconomic. You have a case where you need a vaccine that is actually relatively inexpensive to produce. So, you know, there might be a vaccine that actually exists, but it's very expensive, so it doesn't do very much good in that particular population. So you need to actually design or manufacture something that is cheaper and cheaper to make. Not cheaper in quality, but cheaper to make. And that's where actually good science comes in. I think race and ethnicity has the biggest impact on clinical trials, because there, as Dr. Bash mentioned, what you want to do is you want to have a vaccine that actually works for the people that are going to use it. And there you actually have, and also you have different responses, and they may play a factor in the immune response. So it's more along the clinical lines where it actually has the biggest impact. And of course, we also want representation in people who actually do research on vaccines and who are involved in the manufacture of them.
Thank you. Dr. Bash, did you want to add anything? We move to the next question.
Yeah, I think it's very, there's sort of the immune response question that is important to know. Are there significant differences between different racial groups or ethnicities? And sometimes we do see that and sometimes we don't. I think it was the Haemophilus influenza immunogenicity that tended to be higher in, these are multi-center studies. So the centers where there was a higher proportion of African-American children enrolled, those immune responses as a whole were higher. It doesn't always work that way, but if you don't have diverse populations, you don't have the opportunity to look for it. So there is that question. I'm not aware of any case where the difference is significant enough that you would say this vaccine only works this well in this group of people and it works that well in that group of people. They're much more subtle. And then it becomes important that if you're comparing two groups in the study, for example, that they're relatively well balanced, that kind of thing. So it comes up in a lot of different ways, from the understanding the immune response and making sure that it's broadly effective. But I think on the other side, it's also important because when you participate in studies, then that engenders confidence and and the use of vaccine improving the health of populations. It's the being information that's being generated is relevant. One of the Haemophilus influenza vaccines was really studied in efficacy on the Native American reservations in Arizona, the White River and Navajo reservations. And that happened to be a population that had particularly high rates of these kinds of invasive bacterial diseases in young children, especially very young children. And the vaccine was really dramatically effective, essentially eliminating Haemophilus disease from the reservation. And so there's that entire efficacy trial that was the foundation for that licensure of the vaccine was done in Native American children. And so there was not diversity in that respect, but there were other phase 2 studies that had immunogenicity across a wider range of individuals. So sometimes you narrow in on a population that happens to have a lot of the disease that you're trying to prevent.
Okay, thank you. So we have a question that again is, I think, still linked to the lectures, and it is a really great question. have you ever had any companies who had approval and then changed their vaccine process and lost approval? Has any vaccine ever been compromised by modification, if I'm reading this correctly? Dr. Van, Dr. Bash.
Technically, no. But what has happened is, for example, every time a company makes a change, they actually have to get change in the process. They actually have to get approval for that change. And what can happen is that approval can actually be approval of that change can actually be denied. So the original process is licensed. They want to make a change to that. And we have to approve that change for them to add that to their license. So what gets denied is that you're not allowing them to actually make that change and add it to their license.
Okay, thank you. So the next question I think is COVID specific. So I know you all said that you weren't really here to talk about COVID vaccines in particular. So I will put it on the table and see if you're comfortable in responding. So the question is, it's been reported that we will have to take 2 vaccines, and I think they mean 2 shots or 2 dosages. What will happen if people don't get the second dose? So I know I'm mindful that there are multiple vaccines, but can either of you feel comfortable in speaking to what could be the import of not getting a second dose? Is that one you're comfortable in answering?
And Dr. Sitnar, we perhaps should
Well,
bring
I think,
Dr. Kamangar in now.
Okay, Dr. Kamangar. Right. Yes.
So,
so
Dr.
I
Bash,
was,
responding to this.
well,
Okay, I
the
see. What,
problems of Zoom. I was just going to say that question, but just from the general question, the studies are, clinical studies are designed in a certain way. So the people who were enrolled in this study received vaccine 1 and vaccine the second dose. And, there may be some data that is collected between first and second dose. I can't answer that. I'm not a part of that. I don't have that information. That it's possible that may be discussed on Thursday if there's information about that. But the point is that if the study has been looked at, then the information that people are saying about how well it works has been collected based on this. So the real, the point is I don't know, and I don't think anybody would be able to know exactly what benefit you would derive. Some vaccines do provide benefit after a single dose, and some vaccines really, it's not until that second dose that you really start to see an effect. And so I can't talk to this specifically, but that's kind of the general picture. So you have more to add.
okay, and is that the kind of information for any phase 3 of any trial where that data is being collected and can be shared as the evidence accrues? Would that be accurate? Like that is kind of the kind of information in real life kind of implementation where you can begin to collect that data?
Oh, after licensure, sometimes in phase 4 studies, yes. If there's if there's good surveillance that goes on with the disease, you can sometimes tease that apart.
Okay, thank you. I have a hand raised, but I don't know what to, and Dr. Kamangar, I don't know what to do with a hand raised. But Kashawn, Dr. Wilson, maybe you could put your question in the Q&A box, then we can get your question answered. Because Dr. Ofosu has her hand raised. So Dr. Ofosu, if you can hear me, could you type in the chat? And if you can't type in the chat,
Q&A.
what do we do? Right. She may be on a phone or something and not able to type in
So-
question.
You can ask her to text me the question and I'll relay it to you.
Okay, Dr. Ofosu, Dr. May is saying text it to him and he will read it out loud. If that's okay.
So I know there was some.
Oh, here it is.
questions.
I'm sorry.
Okay. Do you have a
You can
question?
yeah, you can read this one. This is Dr. Ofosu does have something in the chat now. Do you want to handle this one?
Sure. Dr. Foster says, everyone does not have the same level of trust for the vaccines. How will marketing the need and usefulness of the vaccines will be used for the public? So let's say, let me just be a little bit more vocal. There are some people who think Bill Gates is funding these vaccines to control your brain. So we hear all sorts of questions from people. How do we dispel those myths? Is there any way?
Was that a comment or a question?
Yeah.
Oh,
And
well,
I'm happy to add my 2 cents. I'm happy to add my 2 cents on this one, Dr. Bash, Dr. Vann. So I do think this notion of trust that Dr. Bash referred to is critical and essential. So before the meeting, I went through all the sort of horrible, unethical experiments that had taken place and the issues of why some communities may find just a general distrust of a medical system and anything that has the word experiment in it in particular. So I know one of the things we talk about within the school clearly is being able to build relationships with people. So somebody's generally not going to just trust you if there's no reason to trust. So some of the things I think Dr. Fosu in terms of the marketing is not to make marketing just about get the vaccine. The marketing needs to be about education, understanding what the fears and concerns are, and addressing those as we tell people that the vaccine is a good thing to get. So I think it's moving beyond marketing and into what in public health is really social marketing. It goes beyond a product. It is the kind of context, and you have to understand the receiver of the information as well as the message being delivered and shaping that in a way that builds trust. And so I think Dr. Bash and Dr. Vann, actually their presentations tonight are an effort to start building this credibility basis. These are individuals who you can look at, they look human, they look approachable, right? They don't look like Superman and Superwoman, though I know you are. But when you can sort of identify with the people sharing the information, humanize it, I think that is part of the work. And I do think that it's going to take a lot of work and effort. In fact, I just saw an announcement that's going to be coming out, I think from NIH around reaching vaccine messaging in what they usually call, hard to reach communities and populations. So there's going to be a lot of work to be done by social scientists, medical anthropologists, a whole bunch of folks to understand what the barriers are and then to try to overcome them through really clear communication.
Dr. Sitnar, we don't know exactly who's in the audience and who's looking at this on TV. Okay. So if you would introduce yourself and your credentials, and I'd ask that Dr. Kamangar do the same.
Okay, I apologize for those who are not. I mentioned Dr. Mildred Ofosu, who's former member of the Morgan community in our Division of Research and Economic Development. And my name is Kim Dobson-Sidnor. I'm the Dean of the School of Community Health and Policy at Morgan State University. If you want my credentials by training, I have a PhD in public health. Institutions don't matter. I have trained in public health, and then my other background is social science, psychology. And this area of sort of community engagement is the forte we see of the school. And so these are really areas of interest for us. And then, Fareen, you want to introduce yourself a little bit more fully?
Hello, my name is Fareen Kamangar. I'm Assistant Vice President for Research at Morgan State University. By training, I'm a physician and also epidemiologist. And I have been involved with clinical trials and serving on multiple data monitoring committees, etc., for the past 15 years. So I learned a lot from Dr. Bash and Dr. Vann. Thank you very much for your presentation. I think one issue that has been brought up, which is really important, is how much care goes into making a vaccine. From the beginning, that Dr. Vann said, that every single aspect is looked into very, very carefully, making sure that everything is standard, it is repeatable, and it remains the same over the many years. And then when it goes to, you know, after this is manufactured, Dr. Bash took us through that it goes through animal studies. They don't just let this go to human studies without having some evidence. Even human studies, Dr. Bash was telling us that how they start with a small number of people, then they make it bigger, then they make it bigger, so that they get to a point that they feel, oh, this is really safe and effective. And Dr. Bash showed us lots of examples of how these vaccines have substantially reduced rates of diseases and how many lives they have saved, millions in the world over the years. So I have a question for Dr. Vann and Dr. Bash. Dr. Vann, assuming that in a couple of days, FDA advisory board goes well, and a few days after that, FDA approves the vaccine, would you be willing, to take this vaccine, this COVID-19 vaccine?
If I'm in line?
Yes.
Yeah, no, if I'm in line for it, yeah, there's nothing else there.
Thank you. So Dr. Vann, fully approves it, assuming it. How about Dr. Bash? What would you think?
Absolutely. But as I think Dr. Vann was saying, I'm not in the hospitals. I think this idea is more a question, at least initially, of how do you have enough vaccine for the people who are really in the heart of things. But I'll be happy when I'm at that point where I'm in line and I get to get my vaccine. Most of my work right now gets to be done remotely, so I'm not nearly as at risk as the healthcare workers that are in the hospitals. And so that's my biggest concern, really, is for them. But yeah, no, when my turn comes up, I'm there.
So we have two panelists here, one of them a man, one of them a woman, one of them African-American, one of them white. But they both believe that this is a really good thing and they want to take it. And am I surprised? Not really, because I know each of us have our own opinions. But think about it. In 15 years in Afghanistan, 2,400 military men perished. 15 years. Just today, in one day, 3,000 Americans are dying because of COVID-19. So in one day, we see more fatalities in what happened in Afghanistan in 15 years. Why, I will be the first person to take it if FDA approves this, particularly that is 95% effective. So the reason I brought up, this man and woman, is that we are building this vaccine against a, spike protein of a virus. None of us have it. We as humans are very genetically similar. I'm sure I'm very similar to Dr. Vann, 99.9%, and very different from the virus. So I think the vaccine will work for all of us, very effectively, regardless of race and sex, etc. So I think this is what I think. So if I may, yes.
You appear often on Persian radio talking about the pandemic in general, the vaccines and so forth. Are the concerns of the Persian population similar, the same as they are in the U.S. and do they have the same concerns that Black people do, quite frankly?
In some ways, there are concerns all across the world from what I read and I hear from people, because there are many people that there's a lot of misinformation that they are saying these vaccines are built. This whole virus thing is manufactured. And this is a plot to get you to vaccination that controls your brain, etc. So that is rumors everywhere. But in addition to that, people in the third world are much more concerned because they will not be getting the vaccine anytime soon. Dr. Bash and Dr. Vann said that, we will not be getting the vaccine first. Probably December and January, that will go to physicians and those who are critically ill. We'll probably get our turn by April, but when you go to third world countries, they'll probably not get it in a year, maybe not a year and a half. So there are additional concerns there.
Right. So we have this. I don't see any more questions. So I have a kind of comment to your observation and comment. And then I actually have an additional question. And I said this before we began the discussion in our sort of green room meeting. And I want to communicate it here with a larger audience, which is individuals will make their decisions about these choices around the vaccine. And I think it is critical for me as somebody in public health and who wants an informed, critically thinking populace who knows how to kind of take information and make personal decisions. That is, every individual needs to, as the phrase may go, examine his or our own conditions and make those decisions. Of course, we want that decision to be get the vaccine, it's in the public good. But for those who are not there yet, there's nothing wrong with not being there. And there's nothing wrong with wanting more information and asking more questions and going to more sources. Absolutely, positively, nothing wrong with that. And I think that to sort of get back to the other question, this area around trust and credibility comes with communication. So I do think it's important, if we really want this vaccine to reach the levels of implementation when it is widely available to everybody, that we allow people the space to have these conversations without judgment, without anything else going on, and know that we each have our own history that we're dealing with and that we're bringing to the table. The conspiracy theorists, whether it's Black communities who have a history with medical communities of not necessarily being very valued or trusted institutions, you can't just dissipate those because the FDA says this is a good thing, or because even Dr. Fauci, who most people in this environment think of as akin to, Yoda himself in this regard, that we need to realize everybody's not in the same place, everybody does not have the same lived experience, and therefore, if our intent is about vaccination and the public good, that we have some work to do and recognize there's nothing wrong with the work. I mean, we should be putting the work in, and that is the role of public health, I think, in this arena, is not to just create the vaccine, but then to inform, communicate, and assure trust enough so that people want to comfortably take this vaccine and have it have its impact. So I think there's another question, Dr. Kamagar, in the chat box. Do you want to take this one? This is again a COVID one, so.
Question is, how long does the immunity of COVID-19 vaccine take? I hear it takes only 3 to 4 months, meaning it persists for only 3 to 4 months. Is this correct?
I would suggest you tune in to the advisory committee, because that kind of thing would likely get discussed there if there is information.
Dr.
Right, so
Bash?
yeah. Is Dr. Bash, she's still on mute?
Yes, now we can hear you.
Yeah. Hi.
The question
So,
was, is there any evidence of that the immunity
Yeah,
lasts?
sorry about that.
How long
I
it lasts?
can't. Right. So what you have to realize is that these studies are ongoing. So they're doing while the initial emergency use authorization is considered. So the data necessary to answer these questions will, because I actually don't know it, and it wouldn't really be appropriate to discuss this file anyway at this point, but what will be described when they consider this on Thursday will be what's available now. And I imagine that there will be some discussions about when will we know X amount more and when will we know X amount more, or that kind of thing. It wouldn't surprise me. So to kind of feed back on the other comments just a minute ago, it is very hard to trust in a system that hasn't always acted to your benefit or to anyone's benefit, or so that is, I think, a very understandable situation. I think that the more there can be knowledgeable people, trusted people, I do think this conversation on Thursday with the advisory committee, some of it's very technical. Not everybody's going to want to listen to that, but people in the community that do have some background or interest that can listen to this, they can provide their thoughts within their community. And I think that that really helps a lot. You know, there will be, I'm sure, a benefit on the whole once this vaccine is widely available. But in the immediate term, you know, vaccination is going to take some time. So the real benefit is to the person who's vaccinated. And with so many people with conditions that place them at risk for serious disease, I really hope that if the evidence is there, just that it tests people that are at risk for serious disease, that's made clear enough and convincing enough and is presented in a way that's understandable enough that people do feel comfortable so they can protect themselves if that's possible. That's what I really hope for. I kind of hope that some of this background will help the conversations about a specific vaccine, hopefully coming up in just a couple of days, will make more sense. And I think that, I mean, the only other thing that I can convey to people is that I feel I have a privilege of working with very smart and very concerned people. Everybody that I know that works in the vaccine approval process at FDA is concerned with really figuring out those questions. How well does this work? How safe is it? And who does it work in? And I imagine that through this whole time, this struggle of how quick can we go and still be certain enough of all that safety and effectiveness and all of that, this is something that very smart people have been spending all of this year living and breathing and working hard on to make sure that they take enough time that it's safe, but not so much time that people aren't getting the vaccine as soon as they can get the vaccine. And it's not one of these, you know, there's not a black and a white answer or a yes or a no answer. Everything has a little bit of a cost. You know, more safety takes longer. Taking longer means it's not available as soon as it would be otherwise. So the balance is there's a little difference between the balance in the UK, who started vaccinating this week, and here. These are actually, in the big picture, pretty minor differences. This is not, you know, years difference. We're talking days difference here, but there are differences in the shades of gray that making these decisions are so hard. But I work with people, and I feel that I am really lucky to work with people that are smart and doing their best to make those balanced type decisions.
Dr. Bash, would you concur that in most other instances in history, when we have developed vaccines, we followed a much more deliberate protocol? It's taken years to develop these vaccines, but we don't have the luxury of time now with what's going on. So we have developed this quote warp speed process. And quite frankly, the data to address the question that was posed to you is not here now. We would have had that under normal circumstances, but this time it will come out as more people take the thing, and as we get more information from the clinical trials. And I think in our discussion that we had a few days ago when we were prepping for this, someone mentioned that Dr. Fauci had been asked a question like that. That is, how can we do this so much more rapidly and develop a vaccine that's efficacious and safety in a number of months when typically it takes sometimes 10 to 15 years to do? And I think his answer was that in this case, money is no object. All of the money was put on the table to do this, and you got the smartest people on the planet working together to solve this one problem. So that's what's different about this. And that's why it's possible, it has been possible to develop. Of course, they have the messenger RNA, so we've got some new technology also. But that's why it's possible that we can pull this off and do this much, much, much more rapidly than in the past.
There's another question in the
chat. I want to.
I'm sorry.
Oh, can I follow on to that? Because I think you may, and then get to the question. But I think what you just said, Dr. May, is very important. But I want to say that the structure of approval is still there, but it's been condensed because of resources and urgent need. But the actual looking at preclinical data and phase 1 data, that all still happened. So we didn't skip any steps, is what I'm saying, but they could be condensed because of a very focused and big effort. And the vaccine that Dr. Vann talked about for the meningococcal A epidemics in Africa, not to the degree that this has been shortened, but that was an example of a vaccine in which instead of it shortened by many years the time that it took from the initial start of that project to getting a licensed vaccine. And it was partly also because it could be built on the conjugate vaccine technology that people already understood, but also because there was a directed, really focused process to move that along as quickly as possible. And one thing that might allay some people's fears a little bit is that this RNA is there's no licensed vaccine for that, but there has been research on the prior SARS vaccines that are similar. So there was some background to build on, which gave them gave the world a little bit of a head start in terms of developing these vaccines. So I think that also helped. But it's really been the effort, I think, that as Dr. Fauci said.
So may I add something? The previous question that Lale asked was about how long do these vaccines persist? While Dr. Bash and Dr. Vann said we don't know exactly yet, and there will be more information hopefully revealed 2 days from now. and also as studies continue over the next few months, there have been several papers published on this, and there was one that was published a few days ago in New England Journal of Medicine, and they studied 36 people, and they showed that the level of antibodies go up in 30 days. They reach their maximum 30 days, and they stay almost in a plateau for almost 4 months so far that have been studied. So we know that at least 4 months, we will have high levels of antibodies with these vaccines, but we don't know how long after that. It may go on for a couple of years, which we don't know yet.
Okay, so we have two more questions in the chat, 3 more. Want to make sure we get to those, so maybe we can pace our time in answering. First one is, and I'll make this generic as opposed to specific, how do we determine who should receive the vaccine first? So generally, how does the determination made? Is it a general proposition? How you go about deciding who gets the vaccine first, when that decision has to be made? And then how do we get it to the entire how does it then go to an entire population? That's the one question. And then the second question has to do with a comment around the issue of trust. And the third one has to do with a generic question around AIDS. So given those 3, if we can sort of in the next 15 minutes, I guess, pace our way through those questions. So how do we get to choose who gets to be the beneficiary of first batch of vaccine? Dr. Bash, maybe if you're comfortable in a general way, talking about how FDA might approach this discussion.
So,
Or has approached it.
Right.
well, so FDA, this is one of those things that I think is very useful to understand the distinction between FDA's role and CDC's role and state's role, the Vaccine for Children's program. We have like with many other aspects of healthcare in this country, we have a bit of a patchwork quilt. But the role at FDA is to the process of licensure. And it's the data that are generated that determine what we can say about who should receive the vaccine. So generally, if a vaccine's only been studied in individuals from, say, 10 years of age to 25 years of age, that's what we license the vaccine for. It doesn't mean that it won't work in younger people or older people, but that it hasn't been studied in those people yet. And then the question of implementing that information is generally, we have input because we are most familiar with how well it works and how safe it is. But that's where the CDC's role in terms of understanding the epidemiology of the disease that the vaccine's for, who's at highest risk, how prevalent is it? Do we need to vaccinate everybody, or can we have a good effect by just vaccinating a certain group of people? When the HPV vaccine was first licensed, for example, it was only licensed in girls, and now there's a license in boys. And the decision about how do you have the most public health effect from this vaccine. So these are the questions that the CDC and the ACIP in particular, that's the Advisory Committee for Immunization Practices. And they take all these pieces, they take our data, our assessment in, they take the CDC's information in, and they look at where can the most benefit be had and what's the most effective way to deploy a particular vaccine. It's not us who decides that, is the short answer.
Thank you. Dr. Vann, did you want to add anything, or should we move to the next question? You're on mute.
I don't have anything to add.
Okay, great. Dr. Kamagar, do you want to put the next question to the group? You're on mute too, right?
I'm sorry.
That's okay.
The question is, trust is fundamentally based on relationships. It seems to me that potential vaccine recipients should talk with their primary care doc, community health center, healthcare center, pharmacist, family member, or if none of these is available, then anyone they trust. What do you think? That was the question.
So, Dr. Vann, you feel comfortable? And I know we've been wearing Dr. Bash out here. So
I actually am a firm believer in science, and I think, and I hope more people actually become more trustworthy of what happens with science. And I think where people should actually trust, I think they should trust in science and go to persons who actually have that kind of knowledge, if they have them available to address that. So some of the things that are actually mentioned in that piece, I think are good sources.
okay.
I think any good source of scientific information, or even reading it yourself, I think is actually a good approach.
Okay, thank you.
May I pose this question actually to Dr. Sidnor? Dr. Sidnor has been in the community in Baltimore for many years. How do you engender trust when you firmly believe that the science is good and people will benefit from it? And sometimes it is true that you have a distrust, but distrust is a two-edged sword. I mean, sometimes when you distrust a good thing, you may lose an opportunity. How do you engender trust, Dr. Sidnor, in the community, if you really believe this is a good vaccine? I'm just making an assumption.
So the issue is if you're distrustful, then data, unfortunately, as we've come to understand in the past few years, the ability to distort science and facts is problematic. So, Dr. Vann has a background that makes trusting the science as he understands it very simple and easy because he can discern good science. He's got the skills, the tools, the experience to do that kind of differentiation. When you see different people very emphatically telling you these are the facts, and I know them to be the facts, and this is the science behind them, and it's bad science, there's actually room for that distrust to be on the table. So then trust does become a matter of how do I discern what is really the good science, who becomes credible, how do I figure this out for myself? So trust and data is not so, I mean, I love data. I love data, but I also understand how it's created and what people can do with it. And so until you get that comfort level with the data science yourself at some level, or with someone who carries that credibility, then trust remains it remains an issue on the table in the face of a printed sheet that says, as Dr. Bash says, pull out the insert in the pack of the medicine you get, read all about it. Well, I have to believe the process to get me to that pamphlet was appropriate in the first place. And so all of us don't come with that. So that's all, I mean, in that regard, there are layers and there's work for public health to do. I mean, actually, I think that continues to be the work we have to continue to do without cease. So, so the person said, what do you think? I hope he got his answer around that one. There's one more question, unless Dr. Bash has something in particular you really want to add. I want to make sure we get in this last question, which is interesting, but it's an open forum, so it's good to know what people are interested in. Dr. Kamagar, you want to read the last question in the Q&A space? You're back on mute.
So they say, so why haven't they found a cure for AIDS? I guess I shouldn't go there. but these are the questions that people are asking.
They should go there. It's a tough problem.
Mm.
It's not a simple problem. I go to a lot of seminars where people are actually still doing research on how AIDS works, and they come up with something that I think might work, and when they try it, it actually doesn't. So the science behind AIDS is actually very complicated. And that's, sometimes you actually have a disease where it's simple and straightforward, but more often than not, it's more complicated than you think. It's not anybody's fault. The more money you put into it, sometimes it works, sometimes it doesn't.
Okay,
Thank you, Dr. Vann. Dr. Bash, any additional comments?
Oh, I agree completely. It's a really hard problem, and there has been a lot of money in terms of research, but there's also been, I think, a lot of a secondary knowledge that's come out of the basic research that's needed to be done to try and understand HIV. So there have been a lot of benefits that people wouldn't know that have directly come from some of the research, but boy, it's, as Dr. Vann said, people work really hard at this and the answer has not been found yet. A lot of good therapeutic, a lot of good interventions, a lot of good stuff. We're in a whole different place than we were in the 1980s, but there's not a preventive vaccine yet. And I think there's the reports of bone marrow transplant cures, but that's kind of a very rare, the simple answer definitely hasn't been found yet.
Okay.
A lot of people still working on it though.
So thank you. Dr. May, do you want to give any closing comments to the assembled?
Sure. I would like to thank both of our expert speakers for your expert insights and sharing those with us. I'd like to thank our two moderators and the behind the scenes staff, Stacy, Keyshawn, Wanda, for your efforts to bring this to fruition. And most of all, you, the audience, for your participation and timely and pertinent questions and comments.
Yes.
Thank you all very much.
Thank you.
Yes, thank you.
So
Thank you.
Yeah, I would just encourage if folks who can for the public hearing, because there are a lot of questions, and as Dr. Bash and Dr. Vann have pointed out, there'll be a lot of information there, and that may, even if you can't absorb it all, be a good opportunity. I don't know if they're going to be recorded or available live. There are reports and data coming out, and I just want to say that the Morgan State has made a commitment to making sure that we become a center where facts and science and information can be channeled or funneled or made available. So we're going to do our best to work with community and others to make sure that the information that we need to decision make and to protect all of us to be healthy can happen. So I just want to thank everybody. Thank you, Dr. May, for the invitation, and hope everybody stays very safe and healthy over the holidays.